At 3 a.m. your liver is at peak metabolic activity. Not resting. Not idling. Running at maximum capacity, processing the glycogen reserves from the previous day's meals, conducting lipid metabolism, clearing bile, synthesizing proteins that your body needs for the following morning. The liver does not know you are trying to sleep. It does not receive the signal your brain is sending. It is operating on its own clock, set by its own internal timekeeping machinery, executing a program that was written into your cells before you were born. And when that metabolic activity peaks, when the glucose dynamics shift, when the cortisol profile inflects, when the bile acid concentration in the portal vein crosses its early morning threshold, something happens in the neural circuitry connecting the liver to the brain that is not, as most physicians will tell you, a sleep disorder. It is a report. The liver is reporting to the brain at 3 a.m. because 3 a.m. is when the liver has something to say. The science of why this happens is the science of the peripheral circadian clock system, a discovery that has rewritten what we thought we knew about biological time, and which, as of the writing of this, has still not reached the bedside manner of most general practitioners. Jeffrey Hall, Michael Rosbash, and Michael Young won the Nobel Prize in Physiology or Medicine in 2017 for discovering the molecular mechanism of the circadian clock, the interlocking feedback loops of gene expression that generate an approximately 24-hour oscillation in biological activity. The core loop works like this. The clock and BMAL1 proteins bind together to form a transcription factor complex that enters the cell nucleus and activates the transcription of two genes, period, PER1, PER2, PER3, and cryptochrome, Cry1, Cry2. The PER and Cry proteins accumulate in the cytoplasm, eventually re-entering the nucleus and inhibiting the clock, BMAL1 complex, shutting off their own transcription. The PER and Cry proteins then degrade, the inhibition lifts, clock. BMAL1 is free to activate transcription again and the cycle repeats. The full loop takes approximately 24 hours to complete. This is not an approximation or a metaphor. It is a precise molecular oscillator, running in nearly every cell in your body, keeping time through the same principle as a pendulum clock. A self-reinforcing cycle that resists perturbation and maintains period with remarkable accuracy. What Hall, Rosbash, and Young established in Drosophila, and what subsequent work in mammals by Joseph Takahashi at the University of Texas Southwestern Medical Center and Sachidananda Panda at the Salk Institute for Biological Studies in La Jolla, California confirmed and extended, is that this molecular clock is not located only in the brain. It runs in the liver. It runs in the heart. It runs in the lungs, the kidneys, the intestines, the skin, the pancreas, the adrenal glands, and every other organ that has been studied in detail. Every one of these organs has its own copy of the clock, BMAL1 feedback loop. Every one of them is running on its own approximate 24-hour cycle. And crucially, and this is the thing that changes everything, the peripheral clocks in the organs do not necessarily agree with each other, and they do not necessarily agree with the master clock in the brain. The master clock, the one you probably already know about, lives in the suprachiasmatic nucleus of the hypothalamus. The suprachiasmatic nucleus, or SCN, is a paired cluster of approximately 20,000 neurons sitting directly above the optic chiasm, where the optic nerves from both eyes cross. It receives direct retinal input through a dedicated pathway called the retinohypothalamic tract, a projection from a specific subset of retinal ganglion cells that contain their own photopigment, melanopsin, and that respond not to image-forming light but to the integrated intensity of ambient light over time. These cells, the intrinsically photosensitive retinal ganglion cells, or IPRGCs, report the light environment to the SCN continuously, and the SCN uses this information to synchronize the master clock to the 24-hour day-night cycle. The SCN then communicates the time of day to the rest of the body through hormonal signals, primarily cortisol, melatonin, and thyroid-stimulating hormone, and through the autonomic nervous system, which carries timing signals directly to the organs via the sympathetic and parasympathetic nerve fibers that innervate them. This system works beautifully when all the clocks agree. But peripheral clocks are not set only by the SCN's hormonal and neural signals. They are also set, powerfully, directly, and in some cases more powerfully than the SCN, by local environmental signals. For the liver, the dominant Zeitgeber, the German word for time giver, the environmental signal that sets a biological clock, is not light. It is food. The timing of food intake is the primary synchronizing signal for the liver clock. Sachidananda Panda's laboratory established this conclusively in a series of studies using mice, with the critical findings published in Cell in 2012 and in subsequent papers through 2015 and 2019. When mice are given food only during their normal inactive phase, during what would be for a nocturnal animal their rest period, the equivalent of a human eating only at night. The liver clock shifts dramatically relative to the SCN master clock, misaligning by as much as 12 hours. The SCN continues on its light synchronized schedule. The liver is now running half a day out of phase. The two clocks are telling completely different times. And the consequences of this internal misalignment, what chronobiologists call circadian desynchrony or internal misalignment, are not subtle. In pandas mice, circadian desynchrony produced metabolic syndrome, weight gain despite equivalent caloric intake, elevated blood lipids, insulin resistance, and elevated inflammatory markers. In subsequent human studies, including a clinical trial by Pandas Group and a parallel body of work by Krista Verity at the University of Illinois Chicago and Jonathan Johnston at the University of Surrey, similar metabolic consequences were documented in humans who ate outside their body's natural feeding window. But the mechanism by which the liver communicates its state to the brain at 3 a.m., the mechanism behind the 3 a.m. wake-up, is not primarily metabolic dysregulation. It is a direct neural circuit. The liver is innervated by the vagus nerve, the 10th cranial nerve, the wandering nerve, which runs from the brainstem down through the thorax into the abdomen, branching to the heart, the lungs, the stomach, the liver, the small intestine, and beyond. The liver receives both afferent and efferent vagal fibers. The efferent fibers carry signals from the brainstem to the liver, regulating its function. The afferent fibers carry signals from the liver to the brainstem, reporting its state. Winfried Neuhuber at the University of Erlangen-Nuremberg and Gary Boggett-Bott at University College London have detailed the anatomy of hepatic vagal afferents, the sensory fibers running from the liver to the nucleus tractus solitarius in the medulla, which is the primary brainstem integration center for visceral sensory information. These fibers are sensitive to glucose concentration in the portal vein, to bile acid concentration, to osmolarity, to mechanical distension, and to various inflammatory mediators produced by the liver itself. They are not simply monitoring for damage or distress. They are conducting a continuous metabolic status report and transmitting it to the brain in real time. At 3 a.m., or more precisely in the hours between approximately 1 a.m. and 4 a.m. in a person sleeping on a conventional diurnal schedule, the liver is at the peak of its glycogenolytic activity, breaking down stored glycogen to maintain blood glucose through the overnight fast. The glucose dynamics in the portal circulation shift. Bile acid synthesis reaches its daily peak. Bile acids are synthesized in the liver from cholesterol through a pathway regulated by the nuclear receptor FXR and the enzyme CYP7A1, both of which are under direct circadian clock control, with their peak expression occurring in the early morning hours. The liver's circadian gene expression program is at its most active, and the metabolic signals in the portal circulation, the blood supply from the gut to the liver, are at their most distinctive, most unlike any other time of day. The hepatic vagal afferents are receiving a concentration of metabolic signals that is qualitatively different from what they carry at other hours. And those signals are going directly to the nucleus tractus solitarius, which relays them to the locus coeruleus, the primary norepinephrine nucleus in the brain, the main driver of arousal and attention, and to the paraventricular nucleus of the hypothalamus, which regulates the HPA axis and cortisol secretion. Here is the sequence. Between 1 a.m. and 3 a.m., the liver's circadian peak metabolic activity generates a specific portal blood chemistry, elevated bile acids, shifting glucose dynamics, a pattern of cytokine and hormone concentrations that is unique to this window. The hepatic vagal afferents transmit this chemistry to the nucleus tractus solitarius. The NTS relays this information to the locus coeruleus, which increases norepinephrine output. The locus coeruleus projects to the prefrontal cortex, the thalamus, and the amygdala. The norepinephrine signal does not necessarily trigger a full awakening in a healthy person sleeping in alignment with their circadian phase. It is a background signal, a low-level nudge. But in a person whose liver clock has drifted from the master clock, because they eat late, because they drink alcohol regularly in the evening, because they work night shifts, because their sleep timing is inconsistent, because they have non-alcoholic fatty liver disease, which disrupts the liver's circadian gene expression program, the peak metabolic signal from the liver is stronger, differently timed, or both. The locus coeruleus output is sufficient to push the brain past the arousal threshold. The person wakes up. Not because they have a sleep disorder. Because the liver sent a report that was loud enough to be heard. The alcohol connection deserves particular attention, because it is the most commonly overlooked. Alcohol is metabolized by the liver through alcohol dehydrogenase and aldehyde dehydrogenase, producing acetaldehyde and then acetate. Both of these compounds are directly toxic to the liver's circadian clock machinery. Acetaldehyde in particular disrupts the clock, BMAL1 complex, and suppresses the expression of the PER genes, the same genes that are the core timekeeping elements of the cellular clock. A study by Zhong Chen and colleagues at the University of Florida published in Alcohol in 2020 demonstrated that acute and chronic alcohol exposure disrupts hepatic circadian gene expression in a dose-dependent manner, with even moderate alcohol consumption, 2 to 3 standard drinks, producing measurable disruption in the phase and amplitude of the liver's circadian oscillation. The liver clock after alcohol exposure does not tell the correct time. Its metabolic peak shifts. The portal blood chemistry at 3 a.m. is different from what it would be in the absence of alcohol. The vagal afferent signal to the brainstem is accordingly altered. The locus coeruleus is activated at a different phase of the night, or more strongly, or both. The person wakes at 3 a.m., which they often attribute to the alcohol making them sleep lightly, or to dehydration, or to needing to urinate, when what is actually happening is that the liver's disrupted clock is generating an aberrant metabolic report at a time and intensity that crosses the arousal threshold. Now here is where the story changes, because the liver is not the only organ generating these reports. The heart has a circadian clock. The peak expression of the cardiac clock genes, particularly clock, BMAL1, and the cardiac-specific rhythm regulator KLF15, occurs in the early morning hours, and the heart's electrical and mechanical properties vary substantially across the 24-hour cycle. Ariel Bhatt and Darwin Bhatt-Chowdhury at Harvard Medical School have documented the circadian variation in cardiac electrophysiology, the QT interval of the electrocardiogram, the duration of the ventricular repolarization, varies by approximately 20 milliseconds across the day, with its longest value in the early morning hours, precisely when cardiac arrhythmia risk is highest. The peak incidence of ventricular fibrillation, sudden cardiac death, and acute myocardial infarction all occur in the hours between 6 a.m. and noon, the period immediately following the early morning circadian phase when the cardiac clock is most active and the autonomic nervous system is shifting from its overnight parasympathetic dominance toward its daytime sympathetic dominance. The heart's clock is not a curiosity. It is the mechanistic explanation for a mortality pattern that cardiologists have observed for decades without fully understanding. The kidney has a circadian clock that controls glomerular filtration rate, renal blood flow, and the excretion of electrolytes, sodium, potassium, and calcium, all of which vary by a factor of 2 to 3 across the 24-hour day. The kidney's circadian rhythm in sodium excretion, governed by the PER1 gene's regulation of the epithelial sodium channel ENA-C in the collecting duct, is the reason that blood pressure follows a circadian pattern, with its nighttime nadir and its early morning surge. Michelle Haas and colleagues at the Oregon Health and Science University established that disrupting the kidney's circadian clock specifically, in animal models with a collecting duct-specific BMAL1 knockout, produced salt-sensitive hypertension independent of any change in the suprachiasmatic nucleus master clock. The kidney telling the wrong time causes high blood pressure. Not the brain telling the wrong time. The kidney. The gut has its own circadian clock, actually a cascade of clocks running sequentially from the esophagus through the stomach, small intestine, and large intestine, each tuned to the transit time of food through that segment. The gut microbiome has circadian rhythms. The composition of microbial species in the gut changes across the 24-hour day. Bacteroidetes peak in relative abundance during the feeding period, firmicutes during the fasting period, and this microbial circadian rhythm is itself entrainable by feeding time. Aaron Siegel and Aaron Ellenav at the Weizmann Institute of Science published a study in Cell in 2016 showing that the gut microbiome's circadian oscillation is disrupted by jet lag and by irregular eating schedules, and that this microbial disruption correlates with glucose dysregulation. The bacteria keeping time affects the body's metabolism. The bacteria are keeping time. They are part of the clock system. And now I want to think about what happens when these clocks fall out of alignment with each other, because the 3 a.m. wake-up is not the only consequence, and it is not the most serious one. When the master clock in the suprachiasmatic nucleus and the peripheral clocks in the organs are synchronized, when they agree about what time it is, the body's physiological functions are sequenced in a coordinated program that has been refined over hundreds of millions of years. Cortisol peaks at awakening, preparing the body for physical and cognitive activity. Insulin sensitivity is highest in the morning, when glucose from the first meal is most efficiently processed. Gastric acid secretion peaks at midday. Core body temperature reaches its maximum in the late afternoon, coinciding with peak physical performance. Natural killer cell activity, the immune system's frontline defense against viral infection and cancer surveillance, peaks in the early afternoon. Sleep pressure, driven by adenosine accumulation, reaches its maximum in the evening, coinciding with the SCN's melatonin signal, which begins after light exposure ends. Each organ is doing its best work at the time when the whole system has agreed it should be working. When the clocks disagree, when the liver is running 4 hours behind the SCN because of late eating, when the kidney is running 3 hours ahead because of shift work-sleep schedules, when the gut microbiome has a phase that no longer matches any of the organ clocks because of irregular feeding across multiple time zones, the sequence program breaks down. Cortisol peaks at the wrong time relative to insulin sensitivity. Gastric acid production occurs when the stomach is empty or full at the wrong phase. Natural killer cell activity peaks during the sleep window rather than the waking window. The body's carefully choreographed molecular program, which requires temporal coordination between dozens of semi-independent oscillators, begins to execute in the wrong order. The epidemiological evidence for what this does is substantial and, in places, alarming. Frank Shear at Harvard Medical School's Division of Sleep and Circadian Disorders published a study in the Proceedings of the National Academy of Sciences in 2009 demonstrating that a single week of simulated night shift work in healthy volunteers produced metabolic profiles indistinguishable from early prediabetes, elevated glucose, elevated insulin, elevated blood pressure, in individuals with no prior metabolic disease. 1 week. Not years of shift work. 7 days of circadian misalignment in healthy people produced prediabetic blood chemistry. The effect reversed on return to normal schedules, which establishes it as a direct consequence of the clock misalignment rather than accumulated sleep debt. Eva Skernhammer at Harvard Medical School tracked nurses who worked rotating night shifts for more than 15 years and found a 35 to 40% increased risk of colorectal cancer compared to nurses on fixed daytime schedules. The International Agency for Research on Cancer classified shift work that involves circadian disruption as a probable human carcinogen, Group 2A, in 2007, a classification they maintain, based on the convergence of epidemiological data and the mechanistic evidence that circadian disruption suppresses the immune surveillance mechanisms that detect and eliminate premalignant cells. Now here is the part of this story I find worth dwelling on. The suprachiasmatic nucleus was identified as the location of the master circadian clock by Robert Moore and Victor Eichler at the University of Chicago in 1972, and by Friedrich Steffen and Irving Zucker at the University of California, Berkeley in the same year. 2 independent groups, the same finding. Moore and Eichler showed that lesioning the SCN abolished circadian rhythmicity and corticosterone secretion. Steffen and Zucker showed that SCN lesions abolished circadian drinking and locomotor activity in rats. Both papers were published in 1972, and together they established that there was a master clock, that it was in the hypothalamus, and that it was necessary for the organization of biological time. For the next 3 decades, the field operated on a model in which the SCN was the clock and the organs were downstream recipients of its timing signal, slaves to the master. That model was wrong. In 2000, Uli Schibler at the University of Geneva published a study in Science showing that fibroblasts, ordinary connective tissue cells, the least exotic cell type in the body, maintained autonomous circadian oscillations in culture for weeks in the absence of any signal from the SCN or any other neural or hormonal input. The cells kept time on their own. Not approximately. Not roughly. With the same period and precision as the SCN, sustained indefinitely in a dish in the dark with no external cues. Every cell in the body has always been a clock. The SCN is not the master. It is the conductor. It synchronizes clocks that were already running independently. Remove the conductor and the orchestra does not stop. It drifts out of tune. What Schibler's fibroblast experiment revealed, and what the subsequent 2 decades of chronobiology have elaborated in extraordinary detail, is that the circadian system is not a hierarchy with a single master commanding passive slaves. It is a network of semi-autonomous oscillators, each capable of independent timekeeping, each responding to local environmental signals as well as to the SCN's coordinating output, and each capable of drifting from the others when their local signals, feeding time, exercise timing, temperature cycles, social rhythms, light exposure in the organ's microenvironment, differ from the signals the SCN is using to set its own phase. The body is not one clock. It is thousands of clocks distributed through every cell and tissue, attempting to coordinate, succeeding when the environmental signals they each receive are consistent, failing, sometimes catastrophically, when they are not. This changes what the 3 a.m. wake-up means. It is not a broken sleep system. It is a misaligned clock system. The liver's clock and the brain's clock disagree about what time it is. The liver thinks it is metabolic peak hour and is generating a robust set of portal blood chemistry signals that reach the brainstem through the vagal afferent pathway. The brainstem, receiving a signal that would be appropriate if the liver were correct about the time, produces an arousal response. The brain wakes up because the liver's report is credible. The liver is not wrong about its own internal time. It is wrong about what time it is relative to the rest of the body. Every person who wakes at 3 a.m. and lies there with the heart slightly elevated, the thoughts beginning to move, the sense that the body is more awake than it should be at this hour, that person is experiencing a conversation between two biological clocks that are running on different schedules. The liver is speaking. The brain is listening. Neither of them is malfunctioning. They have simply lost track of each other. The solution is not a sleeping pill, which suppresses the brain's response to the liver's report without addressing why the report is being filed at the wrong time. The solution is to resynchronize the clocks, to bring the liver's Zeitgeber back into alignment with the SCN's Zeitgeber, to make the feeding window and the light exposure window and the activity window tell the same story at the same time. The data on time-restricted eating within a consistent 8 to 10 hour window, aligned with the daylight hours, shows exactly this. The liver clock, re-entrained by consistent feeding timing, shifts back into alignment with the SCN. The 3 a.m. report still gets filed. But now it is filed quietly at the correct biological time, below the arousal threshold, while the brain sleeps through it. Go back to the last time you woke at 3 a.m. The racing thoughts that seem to arrive from nowhere. The mild anxiety without content. The sense that the body was preparing for something that the mind had not yet identified. That was not anxiety. That was norepinephrine from the locus coeruleus, activated by the vagal afferent report from the liver, reaching the amygdala and the prefrontal cortex in the pre-dawn hours when cortisol is already beginning its upward climb. The brain interpreted the arousal as psychological. The arousal was physiological. The liver was talking. It has always been talking. You just did not know what language it was speaking.